Practical resources for FMDV sequence analysis. Paolo, the floor is to you.
Thank you. So thank you for giving me the chance to present here. And I apologize with the co-authors of the abstract. I've just reused the template I had, and they are acknowledged at the end of the presentation. I just wanted to summarize basically the report on this work that we have been doing during the last couple of years. Together with the reference lab at Pirbright. We are the basically bioinformatics group, and so we have been building up tools based on the foot-and-mouth disease virus sequence database at Pirbright. Just to give you an idea, of course, as you can imagine, this database of sequences has quite a lot of data. There's been, samples have been collected during the last 30 or 40 years, perhaps even more. And since quite a few years ago, that just used to be VP1 sequences. But recently, thanks to the presence of Illumina and Illumina machine in containment, as you might have heard during other presentations, the reference lab is accumulating more and more data, which includes full capsid sequencing and also whole genome sequencing. So at the moment, I will just focus on this talk on VP1 sequencing, on the database of VP1 sequencing, what we did with it. But all other sequences will eventually enter the picture, and what I'm just presenting is not limited to VP1 sequences. So just to give you an idea, as to the VP1 sequences, there are about 7,000 sequences at the moment, and I just took down as a bit of statistics. So as you see, there are very few, so this collection date, there are very few sequences, as you could expect, that date back to the 60s or the 70s or earlier, but then, of course, there is ramp up in the number of samples and VP1 sequences available for the last years. Also, I just reported this distribution by serotype. So there is a preponderance of O serotype sequences, as you might expect, but also the topotype and so all other classifications, some more detailed classifications have so are not, of course, equally distributed. There are always some kind of topotype or whatever that is dominant. This reflects some kind of sampling bias and also the distribution of different serotypes and topotypes. Basically, starting from 2 years ago, of course, there was realization that we needed tools, both internally and externally. Internally, we need tools to keep track of the samples that arrive at lab and what happens to them, how they're sequenced, when they're collected, and so on. And also, we wanted to make those sequences and information out of the sequences progressively available to a larger and larger public. We have started designing those tools, both, let's say, internal tools that are just used in the lab and external tools that can be used by anyone. And again, the key point is that we are doing that in collaboration with the reference lab. So our goal is that of creating tools that are vetted somehow by the reference lab and provide some kind of expertise embedded in those tools. So as to internal tools, we have this Vibasys, which is essentially custom LIMS, which is laboratory, as you know, information management system to store all information about, again, samples, collection date, sequencing information, sequencing date, and so on and so on. This is a database that can be accessed through a web interface. And basically, it is geared toward generating sequence reports for the reference lab. So one can interactively generate sequence reports and just collect, let's say, batches and other sequences which are present in the database in order to compare the sequences in the batch with the other sequences already present in the database. And also we are working now on the generation of vaccine matching reports, although this is still in the making. So just to give you an idea, the interface to the database is done through a web series of web pages, set of web pages. You can do many things. At the end of the day, you just produce a document which is, again, a sequence report. So sequences which are similar can be listed, and there's all the information about sample and also some kind of tree that shows, again, how the sequence which has been reported relates to other sequences already present in the database. You can also access external resources such as maps or such as NCBI tools and so on in order to make the generation of the report easier. Okay, then as to external tools, we have these FMDV-tools which essentially should provide, again, a part of this information gathered by the reference lab to the public. So they should be globally accessible, let's say. The idea is that, again, they are web tools, so they are accessed through a browser. And, well, I will show you the design. I can show you. So the design is basically very simple. The idea is that this should be accessible from every device which is JavaScript capable. And you can do several things, several workflows that I will show you later in a bit more detail. So you can, again, annotate and classify a sequence. So just put in a sequence, and the system tells you what the sequence is, which kind of genes contains. And again, what are the sequences which are similar to this sequence, and how they are related to the sequence. So it's similar, in fact, to the generation of reports for the lab. But let's say it's more, how can I say, should be easier to use for the general user. And also, we have a geolocation workflow. And finally, we have also a classification workflow that allows you to classify VP1 sequences. So in this case, you can put in any sequence, and there will be a classification. This can be one gene or more than one genes. This workflow is different. It just takes a VP1 sequence and just assigns the sequence to, let's say, serotype, topotype. So it classifies this based on what is already present in the database. So I cannot, unfortunately, give you a live demo because that would not work, of course. So I've just taken down a little bit of screenshots in order to give you an idea. So the first workflow, which is annotation, works like this. Basically, you can either upload your sequence or paste it in this window. Then, okay, there is some kind of CAPTCHA in order to make sure that you are not a robot. And then basically the pipeline just finds out which kind of genes are present in your sequence and generates, so you can select how many similar sequences you want to have in the final tree, in the final report, let's say, similarity threshold, and of course you have to put an email because this takes quite some time to be computed. And then you are, basically the pipeline sends you an email containing a report which has both a list of similar sequences and a tree where you can see all the similar sequences are colored by country, and so that you can appreciate the geographical distribution of similar sequences. Okay, then the second workflow which is implemented is geolocation. Basically, it just shows so each sequence is a point, and it gathers sequences which are close by into clusters. It is responsive, so you have a filter here, and if you just change the parameters, the map modifies just on the fly. So this is very interesting because, for instance, here I was just selecting all sequences from 2010 to 2016. And then I was plotting all serotypes, only the O serotype, A, and Z2 serotypes. And you can see immediately how the distributions are. Okay, of course, you can zoom in and get more resolution. And finally, the third workflow is just what I was saying. So you just put in a VP1 sequence, and so the interface will launch some computation which will classify the sequence and tell you what it is up to, let's say, the sublineage level if there is sufficient information, or up to the level which is available if there is not enough information. And so I just want to thank Luca and Jan. So Jan has been the father of Vibasys. Luca has been collaborating with me to implement those FMDV tools. And then, of course, several people at Reference Lab have been very helpful with everything and very stimulating as well. Those tools should be available soon from the website of the Reference Lab. We didn't put link in yet, but we will do that let's say when we're back, basically. Thank you.
Thank you very much, Paolo. Impressive, and I appreciate that there's a lot of work behind all of this. Questions? Over there, yeah.
Thank you, Paolo. We have been waiting for this. I understand that all the references, all the sequences which are generated at Pirbright will be in this site. But what about the sequences which are generated and which are submitted to the public database? Will they be included as well?
Yeah, so I think the idea is to have all sequences Pirbright and DOL also the one publicly available, so the ones from NCBI and so on. That is something that we will eventually do. So.
But I think, of course, a key advantage of this system that we imagine is that if you go into GenBank, there's very little expert curation of those sequences. So I think those of us that look at GenBank a lot, you will see there's a lot of mistakes, a lot of errors. And the hope is that having a single place that is FMD specific will correct a lot of those errors. And we can have data that between the community, we can discuss whether it is right or wrong, and we can correct it ourselves. And hopefully that will make some of these analyses a little bit more robust. There's another question.
Yes, did I get properly that we will have access to all your 7,000 sequences? Will it be available to the general public?
Well, so this is, I cannot answer this question, but at the moment the sequences cannot be accessed, but you can access, so they are used, let's say, by the pipelines, but you cannot download them. But this, again, is not my, so this is what I've been told to do and what I did.
And and we can also upload our sequences in your system and see where it fits in your trees. That's
right. And also you can get, let's say, the metadata information out of like geolocation and these kind of things.
Thank you.
And very much the design of this system is really to be shared amongst the network of laboratories. So I will say something towards the end of this session about the work that is going on within the OIE FAO network. And this is a tool that is not just a Pirbright specific tool, it is for all of us to use, but it comes as, I mean, I think we should discuss that later, but it will come a little bit with some certain constraints. So we cannot make all the data freely accessible to everybody, because there will maybe be the possibility that people just use the data and use it for other purposes. So I think, I mean, that would be a subject that I would like us to think about maybe for the next half hour or so, and when we come back to it, we can discuss under what sorts of arrangements should we make the data freely accessible.
Okay, thank you. Last practical question then. Paolo, what is soon? When will the FMDV tools be available? Can you give us a date?
You mean, so basically everything is working. We only have to do a technical thing, which is making sure that there are not many concurrent accesses to limit somehow the computer. But the site is fully working, so it's just a matter of a few days.
Days, okay.
Yeah, no, seriously. No, everything is working fine. So we just have to limit the number of concurrent users.
Okay, we'll give you 2 weeks.
Okay.
To
More
be generous. Thank you very much.